across the India have joined for this session, right? So, and I hope everyone gets some of the message from this presentation. Even many of the participants having much experience, and they are teaching a number of students in their academia, right? So maybe some of the new learning from this session. So I will share my personal experience, whatever I had completed in 12-year industrial trainer, right? So as Dr. Devesh brief about me, it's a nice pleasure, and definitely my talk on sterile formulation development, what are the development challenges and manufacturing challenges, and what are the global regulatory expectations for different regulated markets. Right. So in my talk, I will cover these are the aspects, different development strategy, selection and optimization of excipient, quality concepts, and definitely I will also explain one of the case study on lyophilized product. So everyone knows whatever the substance we use as a drug or dietary supplement, definitely they have some of the therapeutic indication. So with respect to the quality, safety, efficacy, definitely every drug molecule or substance should be pre-screened by the different quality parameters as well as safety and efficacy. Then we can claim it's as a medicinal or a dietary supplement. So what are the differences in academic research and industrial research, right? So as per my understanding, the major difference is end user. In academia, we don't have a end user application, but in some of the pioneer academia like NIPER or many of the pioneer or reputed institution having industrial collaboration for the different projects which are running in collaboration with the industry and academy program, right? Dr. Davis, am I audible to everyone, right?
Yes, sir. Yes, sir. Yes, sir. You are very much audible, sir. Yes, sir.
Yes, yes.
And your screen is also visible, sir.
Yes.
Okay.
So here I given a brief idea about the development strategy, right? How we have to design our development of any new drug product or new drug formulation or new drug substance, right? So these three are the major section we have, like critical evaluation. And even we know people having different thoughts in their mind, but only the very few thoughts can be executed at the shop floor, and anyone can give the genius idea that can be implemented for commercial application or partial commercial application. There may be different approaches, right? So for the development program, I just here I'll highlight without strategy, it's like a ship without a rudder, right? So before starting any development of any drug substance, formulation or drug product, whatever, we have to have proper strategy for the development from the initial means kickoff of the development from the till successful commercialization. I will share one of my good experience. I had completed my doctorate in nanopharmaceutical technology, and the formulation was ophthalmic nanoemulsion. And proudly I can say, on current days, it's more than 25 countries. It is successfully commercialized, and we have credit of good commercial demand from the different regulated markets. So before starting any development strategy, we should have to properly evaluate all the aspects, whether it's a IP protected, means already someone have already patent on this product, or same formulation, same composition. What are the they have claimed in their patent? What are the manufacturing strategy they adopted, and what is our backup and follow-up plan for that particular research, right? So, and what is our target product profile, and how much share we can take in next coming future when our product get commercialized in the market, and what are the different regulatory challenges, what are the different regulatory expectations to file our drug product in that particular market. So these evaluation we need to have properly evaluate before starting the development. Then early stage evaluation means, suppose we already have evaluated this any formulation, but before going forward, we should have to evaluate the proper API supplier, proper manufacturing facility feasibility, available resources, cost optimization. Means we cannot use any of the excipient, we cannot use any of the method for the development. So before implementation or execution, everything should be very clear on the paper. And in industry, we generally have 3 or 4 presentation during the development. First is initial kickoff or initial evaluation by the different stakeholders, like business development, market survey, and marketing team, regulatory team, formulation team, analytical team, manufacturing team. So different stakeholders take place, and they give their views. What are the challenges? How we can make it feasible for the next coming 24 months or whatever the timeline we have decided to launch that particular product. Next is pre-formulation evaluation. So definitely most of the activity we perform in both the platform, means academia and industrial, right? Like polymorphism, everyone knows. In some of the formulation, if we review the literature properly, so in some cases, I just give you one example. There is a product, fluconazole injection, also have an official monograph in USP, BP, IP, in multi-compendia, and the same formulation available with the sodium chloride as well as dextrose. But the concern is there are 3 different polymorphic form of fluconazole, but only the form 3 is more stable in compared to form 2 or 1. And in pharmacopoeial monograph, they also given the different analytical methods, method 1 and 2 or 3 or 4. So according to the formulation, according to the route of synthesis of the API, suitable method to be applied for the analysis. Again, force degradation of the API and other parameter as well. So we need to evaluate these such pre-formulation evaluation during the early stage. When we talk about the parenteral, right? So other than the formulation, means which are administered other than the topical route, or we can say other than the oral route, which considered under as a parenteral. Topical we can not directly say, because ophthalmic formulation also come under the topical formulation, and that is the sterile formulation, right? So majorly parenteral we divide in two parts based on the volume. Up to 100 ml we consider as a small volume, and 100 or above 100 ml we consider as a large volume parenteral. So major variety in, if we see in SBP only, but in LVP we have very limited variant like infusion or ready use solution for IV bolus or intravenous infusion. And with respect to the formulation variation, like dry powder, lyophilized powder, emulsion, suspension, with respect to the container closure system like pre-filled syringe, vial, ampule, ampules, plastic containers, LDP, SDP containers, means different MOC, different packaging style. We have multiple variants in small volume parenterals only. Even with respect to the administration, like respules, we can administer as a respiratory solution. It is also a sterile formulation. Ophthalmics, that is also a sterile formulation. And definitely injection also covers in this when we spoke on parenteral. But in parenteral formulation or sterile formulation, we have very limited scope of the excipients, which is approved by the different regulatories, right? So major category I will explain here. First, definitely we have the active pharmaceutical ingredient which gives therapeutic effect for particular target disease. Second, we can say solvent or vehicle. So major of the sterile formulation having the water for injection. But apart from the water for injection, we also have some of the formulation with the hydroalcoholic base, like in some of the formulation like cabazitaxel, having ethanol as a solubilizer and a diluent as well, which is diluted before the administration, and after further dilution, it can be administered to the patient. Like PG and other solvents are there. Preservatives, so number of preservatives are there, and most common preservative benzyl alcohol in injection cases, and benzalkonium chloride in ophthalmic cases, and some of the parabens also used. But parabens having the major disadvantage of degradation during the product life cycle. So we need to add some overages at the time of formulation manufacturing, maybe 10% to 20%. And BKC having the beauty, it is only used at point not 2% as a standard recommended concentration for ophthalmics. In most of the formulation, if you see in the market, that is having only the point not 2%, or maybe less, or quite higher. But if you see in next slide, I will give you the detail where we can find out the approved concentration, what we can use in that particular formulation or route of administration or dosage form. So I will also give you the brief idea. Next, solubilizing agent, tonicity agent. So everyone knows tonicity. Generally, we can monitor, contribute from the sodium chloride, propylene glycol, glycerine, mannitol, dextrose. Number of tonicity agents are there. Buffers, definitely everyone I think knows phosphate acetate, borate, citrate. Different buffers are there which we generally use to maintain the pH. And stabilizer, some of the formulation also having, like sodium metabisulfite, antioxidant, chelating agent, complexing agent. EDTA is the most common example for that. So it's a common manufacturing process flow chart for a simple solution for injection kind of formulation. So this is like initially we collect the WFI in a final compounding vessel or manufacturing tank. Then we dissolve the respective excipients before addition of the API. And I will share here one of the good achievement in one of my patent I have. So in that formulation, generally in my formulation scientists having the mindset before we generally add the excipient before addition of API. But I have one of the tricky approach on which is patented. So I just have the trial in 10 year back in the lab. So I found some of the precipitation when we dissolve the excipient before addition of API. So when we added this API, so suddenly I found API gets precipitated. So that time I thought just to have the dissolve this API before addition of excipient, and that approach gets work out, and luckily I found a patent for that. So it's not a thumb rule. We have to add and dissolve the excipient before addition of the API. So it totally depends on the order of mixing and your chemical uh, quality parameter based on the respective analysis of excipient as well as APIs, right? So during the formulation design, we also have to monitor and freeze the order of mixing. So that can be followed in the manufacturing process at the plant. Then after API addition, we have to check the in-process bulk, pH, then filtration through sterile grade filtration, and in-process bulk analysis release. Right? So it's a common manufacturing process. Now, what are the challenges we generally face during the development? Some of the participants having a good idea about the sterile formulation. In sterile formulation, US regulatory have a strict regulation, means whoever the develop this generic formulation of any product, so that should be pharmaceutically equivalent. Means Q1, Q2 formulation should be same as the innovator. Q1 means qualitative and Q2 is quantitative. Composition should be same with the innovator. Limited scope of excipient, as I told in previous slide. So here is the one, just a moment. So if you see, this is the web link where you can search the excipient, what are which is active and hello, Dr. Davis, can you see my screen, right? Hello.
No, sir, it is not visible. It's not visible. Yeah.
Hello. Just a minute.
Not visible.
Is it? Yeah.
Now only it is.
Yes.
You are
Yes.
sharing.
So this is the
I-
web link where we can find out the respective excipient quantity for respective dosage form. How much quantity we can use in our respective formulation, like oral route and capsule formulation, right? So we can here screen this respective excipient for respective dosage form and respective route of administration, and we can find out here the maximum potency per unit dose, like 0.2% of the daily intake. So I just here giving one example of povidone K30, which is generally used as a crystal inhibitor for the sterile formulation, in mainly in injection and ophthalmic formulation. I just give you one example, which is having this olopatadine in 0.7%. It contains about 0.2% of the povidone K30 in the formulation, which act as a crystal inhibitor. So here we can find out the respective excipients, right? And one more thing I will share. And this is the website of FDA, where we can find out the reference listed drug in US. Like I just sharing here one of the. I will also explain about this product in my next coming slide. This is azacitidine, right? Here we can find out the reference listed drug in US market. So as I briefed about the excipient, where we can find out the respective formulation, respective quantity, respective dosage form, right? So this is the web link where we can find out, as I explained. And next is compatibility, right? So in the case of injection, there is no drug excipient compatibility required. When we talk about the same Q1, Q2, this information you can also can see in the FOQ of the FDA database. But in the ophthalmic case or other like some of the complex molecule, where we require this compatibility with the excipient filters, and in injection case, filters, in process material like tubing, diluent, and container closure system. These selection also very important during the development. Container closure system generally we consider for the glass vials, plastic bottles in 3-piece or 2-piece plastic bottle, flexi bags like NPVC, PVC, and LDP plastic container for injectables also. Selection of sterilization method, which formulation scientists first apply the common approach for sterilization. That means 121 for 15 minutes. That is a common approach, but it's not a thumb rule for all the product, all the formulation in respect to the packaging style. Some of the formulation are thermosensitive, so we need to evaluate the respective sterilization cycle at different temperature and time condition, and that should be valid. That method should be validated properly with respect to the F0 value and other regulatory parameters. In microbiological evaluation in sterile formulation, we generally consider 4 of the parameters, which is like microbial enumeration test, bioburden, sterility, and bacterial endotoxin test. And sometimes we also consider the particulate matter for subvisible particles. Like, so for the subvisible particle, as per the USP General Chapter 788 and 789, we have the criteria for small volume parental and large volume parental. Like for large volume parental, they have a specification limit for, and they also given the two methods for evaluation. One is light obscuration method and second is microscopic method. So generally for heterogeneous system like colloidal solution suspension, we use this microscopic method, means hot stage microscopy we generally apply. And for light obscuration method, we generally apply for the homogeneous system like true solution or same solution like kind of formulation. So in SVP, they have a acceptance criteria 6,000 particle of 10 micron or less than 10 micron, and 600 particles per container for 25 micron or less. And in LVP case, this is a different acceptance criteria, means 25 particles of 10 micron or less, and 3 particles per ml for 25 microns. So these are the general acceptance criteria given in the USP General Chapter 788 and 789 for ophthalmics. Manufacturing process viability. So as I told, during the development, we have to evaluate the order of mixing, temperature, process temperature, pH condition, and so on. So these different approaches we need to evaluate during the development for smooth execution at the manufacturing shop floor. And with respect to the validation of analytical method, sterilization method, filter validation for the regulatory requirement, as well as robust method application for the respective analysis and respective process. And next is stability. Stability is a very, very important and critical for the any drug formulation. At what time, what shelf life we can design, we can propose, at what control temperature condition, recommended storage condition we can propose for that respective product. With respect to the manufacturing challenges, definitely controlled classified areas should require for the sterile formulation, as everyone knows. Heating, ventilating, air conditioning system, environmental monitoring, like static monitoring, and microwave plate exposure at respective area of the manufacturing. Periodic media fill validation, like most of the regulatory criteria having a biannual media fill validation. Equipment validation means predefined schedule equipment validation should require. Whatever the utility and operation cost in case of liquid formulation, definitely utility cost is not much higher in compared to the lyophilized formulation, because not only lyophilization, as well as some of the complex formulation like suspension or microsphere kind of product that having a high utility and operation cost. And area qualification, equipment qualification, that also required for the regulatory requirement. And process validation, like 3 validation process validation that we need to manufacture before commercialization of the product for the respective market. That is the regulatory requirement. Now it's about the filtration trend. How we design, how we will select the suitable filter for our formulation. So that is a filterability trial study, right? So here is one of the brief case study. Different material of construction of the membrane of filter are available in the market. In earlier days, most of the people are using nylon filter. Hello.
Yes, sir. Continue. Maybe there is a little disturbance.
Okay. If anyone having some comments or queries, so they can raise their hand, or I will address their query in completion of my presentation. So here different material of construction of the membrane filters and cartridge filters are there. So in earlier days, as I told, nylon filters were very easily available and most applicable for the pharmaceutical formulation. Even nowadays, also in lab application, it is continuing, but it is having some of the limitation that nylon membrane cannot be utilized for the higher pH side or more throughput of the product for the low surface area. So then polyvinyl dyeing filter came in the picture in the decade of 90. Then uh, in the 2000 they introduced new membrane polyether sulfone. So I just give you a brief about the polyvinyl diene and polyether sulfone comparative. So PVDF filter can be applied for the formulation which is having pH 2 to 10, while PES filter can accommodate the formulation which is having pH 2 to 14. So that cover a broad range in compared to the PVDF, right? And PES definitely having high throughput when compared to the other membranes. So definitely when where we can save the time for industry. PDA chapter 26. Sterile filter we generally consider for the pore rating 2 or less than 0.2 micron. That comes under the sterile grade filtration, and it's a absolute filter. And our this pore rating, most of the filter having nominal filter. So these are the common differences in different MOC and different pore rating and other parameters of the different filters. So some of the primary and some of the additional, or we can say secondary quality parameter which we need to monitor during the formulation in sterile, like description, we know description, identification, pH, these all are the common parameter which we need to monitor and control in the any sterile formulation. But based on the nature of the product, like emulsion, suspension, dry powder, lyophilized powder, prefilled syringes, uh, what kind of formulation, what kind of containers are used in the formulation presentation. So depend on that, we have to some additional parameter. Like if I say for intravenous injection and for ophthalmics, osmolality is mandatory. So we have to have control this parameter. For emulsion, suspension, and pre-filled syringes, viscosity is very, very important. With respect to the regulatory market, like Canada, surface tension is mandatory parameter for all the liquid formulation, liquid sterile formulation. In ophthalmic case, drop size is very, very important with respect to the doses delivery. Means every drop count having respective dose of the particular API, means particular dose which we require to prevent or cure that particular disease. So drop, individual drop having respective quantity of the API. That's the reason to control the drop size. Water loss for semi-permeable container like LDP bottles and flexi bag like NPVC bags. And in case of glass container, water loss study, water permeability study is not required. Dissolution study in case of emulsion, suspension, and lyophilized liposomal formulation, dissolution study is required. Particle size distribution for suspension case and zeta potential definitely, and droplet size distribution for emulsion or nano emulsion, micro emulsion kind of formulation. So these are some of the additional parameter depend on the nature of formulation. Need to apply as a quality parameter to control its criteria. How we can control and monitor the quality of drug product? So in the year 2008, FDA introduced and accepted this quality by design approach for the pharmaceutical formulation. And in the year 2012, they implemented a draft guideline, and from the 2015, they forcibly implemented from July 2015, and no dossier after the December 2012, 2015 accepted without application of QVD trials in the development of any new drug product. So the concept given by the Joseph M. Juran, a systematic approach of the drug development that began with predefined objective, means whatever the objective we have for the formulation, that should be predefined, and proper approach means most feasible approach should be applied for the control and feasibility of the manufacturing by evaluating the risk assessment of that formulation. What are the different merits to apply this QVD concept during the development? So that's the beauty. We can save the time, we can save the cost, we can have a good quality of product when we apply this QVD approach for the development. So these are the different criteria, or we can say advantages, which we can find out after having a quality product when we apply this QVD approach. So as you know, in QVD generally we have a quality target product profile that should be predefined before the development. Based on that knowledge is different knowledge aspects, like as I shared inactive ingredient, where we can find out the concentration, and so this is experience and knowledge which we can apply for the design of formulation, and we can also identify the critical material attributes, process parameters, that gives a quality parameter, and that parameter we need to monitor during the stability of the formulation and drug product. So in a simple word, we can say it's a scientific, risk-based, holistic and proactive approach for pharmaceutical development. So these are the different elements of QVD. Like, as I told, quality target product profile, critical quality attribute, risk assessment, design space, control strategy, and process improvement. So here is one example of QTPP of a lyophilized product, right? Suppose if our objective is to develop a generic formulation of X product, which is available in a lyophilized form. So such of the parameter we need to define in a QTPP, like a strength, formulation, container closure system. What kind of container closure? Like if they use a glass vial. In glass vial, we generally know two kind of glass vials are available. Means clear or flint, we can say, glass vial, and amber glass vial. Based on the thermal coefficient, there is a three type of glass generally used in pharmaceutical, and in parental formulation, type 1 glass only used for the liquid formulation, and type 2 and 3 sometimes we can use for solvent base or dry powder kind of formulation. route of administration, appearance, fill volume. These are the different parameters. And when we talk about the quality parameters, so like reconstitution time, water content. Reconstitution time is very, very important in lyophilized kind of formulation, because some of the critical aspect in a unevaluated or kind of formulation means we can scientist can easily manufacture the lyophilized formulation, but it doesn't mean that can be reconstituted in a 5 minute or 10 minute or so on, right? So it's having a better acceptance criteria, means formulation drug product should be reconstituted within a 1/22 or less than that, because paramedical staff cannot reconstitute the formulation for a longer time. And to control the impurities, or we can say degradation product, lyophilized formulation having the minimum water content, that will help us to maintain the quality of product for a longer time. So these are some of the quality parameters which we need to define in a QTPP. And this is about the risk assessment of CQA of the drug substance as well as drug product. With respect to the, if you see this, so here I have described the drug product critical quality attributes, and here the drug substance, right? So if we see, the appearance is very, very important. Means if we have a white crystalline powder at the initial, so that should be maintained during the shelf life of the product. And if it turns into a light yellow, pale yellow, or little bit blackish particle in the description, so that can be rejected at any time of the. There may be a lot of the reason, maybe some of the quality concern, some of the contamination during the production, or during the packaging process, or whatever, right? So these are the different parameters which we need to evaluate in respect to the formulation as well as drug substance. So this assessment need to be done before execution of the activity. Design space. There is a linkage between material attribute, process parameter, and quality attributes. We cannot deliver a quality product without assessment of material and process. So we know there are three rings like control space, right? So for example, if we see the IH database, and most of the person knows 0.9% sodium chloride gives isotonic formulation, but it doesn't mean every time we need to use 0.9%. It depends on the formulation and other excipient osmolar tonicity contribution, right? So we know the control space may be 0.3%, design space may be 0.5%, and we know up to 0.9% we can use. So this kind of linkage between the material attributes and process parameter, that gives quality product. So the control strategy, generally we cover product specification, procedural control. As I told, manufacturing process we have to evaluate during the development, means order of mixing, temperature, time. These all parameters we need to evaluate during the development. And this is about the life cycle management and continuous improvement, how we can cater our product for a longer time in a respective market, how we can maintain our USP in the market. Some of the formulation still continue in the market, like if you see the leuprolide acetate, comes in a microbial microsphere formulation. It's still continue in the market since 1960. It's almost more than, we can say, 50-year journey in the market, and still it is a quality product. Even they introduce only first time they introduced 3 days depot formulation. Now they have up to 6 months depot formulation in the market. So the quality parameter is very important for any drug product, and all the quality QBD approach and their elements need to be implemented during the development as well as for successful commercialization to register the product, retain the product in a market for a longer time. So now the case study, I will explain about one lyophilized formulation. So as we know, these are the major steps in a lyophilized formulation. Solution filled in a vial, like, so first we make the bulk solution that come in a freezing, is then primary drying, secondary drying, and finally we got the freeze-dried product, lyophilized product. So as we know, the lyophilized formulation generally used for the removal of water, which is means those kind of drug molecule which is not stable in a liquid state or aqueous formulation for a longer time. So this approach we generally apply for those kind of molecule. And 3 different stages are there: freezing, primary drying, sublimation, and desorption. Some of the objective, as I told, for extension of shelf life of the product or enhancing the stability of the product. So these are some of the we can say different observation we found during the lyophilization cycle, like melt back, collapse temperature. These problems happens due to the lacking in the development evaluation. Means some of the scientists directly design the lyo recipe without knowing the glass transition temperature of the drug molecule. So some of the problems occurs during the lyophilization, and it's an ideal cake if you see, right? So if we know about the all quality aspect and different we can say characteristic of the drug substance as well as cryoprotected material, so we can design a proper lyophilization cycle. These are the again some of the limitation. Means we cannot design a lyophilization cycle for a 1 month or like that. So still, as I know, maximum lyophilization cycle are for the commercial market is about 5 days. Means 120 hours or 116 hours maximum lyophilization cycle I have seen for one of the product, and minimum lyophilization cycle we found 7 to 8 hours. And these are the ideal characteristics, as I told: intact cake, sufficient strength, uniform color, sufficiently porous. Means it's a aseptic product, so we cannot claim as a terminal sterilized product. So lyophilized formulation generally we filter 0.2 micron, then introduce in a lyophilizer for final formulation. So these are the different formulation components of any lyophilized product, and these are the different steps of lyophilization. Initial freezing, annealing to release the stress of cake. Final freezing, primary drying, secondary drying, then final stopping and unloading of the product container from the lyophilizer. And this is one of the example of one product lyophilization recipe. And during the ongoing lyophilization cycle, we can monitor the product temperature with respect to the self temperature and other parameters. So we can extend or reduce the lyophilization cycle in ongoing cycle. If suppose sometimes it may happen to see the cake is not properly dried, so that time we can extend the whole time in primary drying or in secondary drying. That can be possible in ongoing cycle. And this is a stability of one product. For up to 6 months, we have monitor in this product. And here I am not presenting a whole stability of that particular product. I just giving an example. We generally perform the stability at different storage condition, like for a storage accelerated 40-75. Long term, definitely we have depend on the zones, like 30-65, 30-75, and 20-65, 25-60, right? And these are the common example of the marketed formulation. And definitely some of the formulation which is developed by me successfully marketed in a different regulatory market as well as in Indian market. Some of you can find easily in the Indian market as well. I can say proudly this is daptomycin, which is earlier introduced as a cold chain product, means storage condition was the 2 to 8. Now it's a room temperature stable product at 25. Same way in rabiprazole sodium for injection. And I also have one patent on azacitidine, so which is successfully commercialized in the market. Cyclosporin ophthalmic microemulsion, we can say, that is also a patented formulation from my hand. And dapomedrol acetate injectable suspension is a RLD, but I have developed this generic formulation. Clevidipine and dibutyrate injectable emulsion. So these are the some of the common example which is which we can see easily in the Indian as well as international market. And the different regulatory requirements. So nowadays most of the regulatory agency asking about the extractable leachable and container closure compatibility system, depending on the different general chapter and different quality guidelines by recommended by the agency, like Product Quality Research Institute for Extractable Leachable, ICH, and pharmacopoeial forums. So in a summary, we can say product development, process development, and container closure system is very, very important during the development and designing of any new formulation or new product or a new drug substance. And these are the some of the useful web links which you can use for a quick reference and find out the different information about the formulation as well as drug substance or analytical method like dissolution method, media, and other things. So probably it will be helpful for you all. That's it. So I think this talk given us such message on development as well as other aspect of the development of formulation, substance, or drug product.
So thank you so much, Dr. Kashyap, and I would like to request participants if they have any queries, so they can ask their queries, or also they can put their queries into the chat box. So kindly raise your queries if anybody is having any queries, please. Okay, once again, thank you so much, Dr. Kashyap, for this fruitful session on development, manufacturing, and regulatory requirement for the sterile product. Sir has given a detailed idea regarding what kind of the regulatory requirements and like what are the minute things we can keep into the mind when we are doing a product development, specifically regarding sterile formulations. So I'd like to thank Dr. Kashyap for this fruitful session. Thank you so much, sir. Thanks for your valuable time. Thank you so much, sir. Thank you.
Thank you, every participants, and especially for AICTE and Ram Patel College of Pharmacy to giving this interactive session. Hope every participant enjoy this session and learn something from this session. And this session may be useful for the new projects which are going on in your academia or in upcoming projects as well.
Okay. Thank you. Thank you, sir. Thank you so much. And we will join.